Good morning. Let's get started. Looks like it's just a couple of us for now, but it is 8:30, so we will go right ahead and start. Thank you for being on time. Why don't we do a quick recap of what we've learned so far in the lesson? So previously, we were talking about the replication bubble. Okay, so here is a replication bubble. And we talked about several enzymes that are going to be involved in this process. We have the helicase here that is going to be unzipping the DNA. Uh, it was drawn like this, right? The helicase here, which helps unzip the DNA, creating the bubble. We have the gyrase that are further down that would help relieve the tension. We have these single strand binding proteins that would hold the two strands apart. Um, and we learn about how there are directionality associated with each of the DNA strands. So if the top strand is 5' to 3', the bottom strand must be 3' to 5'. Okay. So I'm not going to redraw all the enzymes. Hopefully you have the note that you took from last class and you are able to refer to it as I'm talking through it. Right, so we do have the helicase here to unzip it. Now, the main enzyme that is responsible for creating the new strand, that is called the DNA polymerase 3. Right? But DNA polymerase 3 has two problems, right? Number one, it's not able to begin the process. It doesn't know where to start. And number two, it can only build in the 5' to 3' direction on the new DNA strand. So to solve the problem where it cannot start, you might remember that we have to put a little bit of an RNA primer. RNA primer created by RNA primase. Okay, primase is the enzyme that creates the primer. Okay, so here is primer. Primer is just a nucleotide, right? The ACTG. Let me show you from last class here. The ACTG stuff here, right? Just a few of them, right? And because it's RNA, there's no T. We are using the uracil instead. The whole purpose of having a primer is to help position the DNA polymerase 3. Okay. Now take a look at the numbers here. This is 5 to 3, which means when I'm building a new strand, it is the other way around, right? So when the DNA polymerase comes, it will build upon the RNA primer. It will continue on from where the primer left off. And because it can only build from 5' to 3' direction on a new strand, it is going to go this way. All right. Now, if you put a primer here at the bottom, same story. Look at the number. This is 3. So that must be opposite, 5 to 3. And this time it is going to go to the right. Like that. So I'm going to remove the helicase here for a second. What happens is when the helicase opens up more of the bubble, the top strand is able to continue to keep copying until the very end, right? So this top strand that is being synthesized in continuously in one single strand with no break, you might remember that's called a leading strand. Okay? But at the bottom, as you open up more, you're gonna have to put another primer and another primer, right? And then another primer. And so we will be creating little fragments. Right? So the bottom strand is not being synthesized in one piece. You do it a little bit at a time. So that's called the lagging strand. And these fragments, right? These fragments, they are called Okazaki fragments. Okay. Now, if you switch your focus to the other side of the fork, right, the helicase here will open up more, and what happens is the lagging strand that was on the left, actually this is able to continue all the way to the end. Whereas the top strand now, like I'm gonna need to have more primer, right? So actually, the leading strand on one side of the fork becomes the lagging strand on the other side of the fork, whereas the lagging strand at the bottom will become the leading strand on the other side of the fork. I'm just going to pause for a moment to see if you guys have any questions. All right. Now let's take a look at the rest of the story. I'm going to focus on this part here. Okay, I'm going to zoom in here a little bit and see what happens. So like we have the strand here, the original strand, and I'm just gonna again make up some letters here. Okay, and then more letters here. Actually, you know what? I'm gonna make a few more here. Right. So I'm looking at the bottom strand here, which means it's 3' to 5' here. Now we already know at the beginning there is gonna be some primer there, right? So the primer is going to look like this. This is A, which we're going to put U. The primase is going to put the U, A, G, C like that. That is the primer. And then you know, we're going to have another primer here. So the DNA polymerase 3 will build the new strand. It'll continue to build, right? It'll continue to go forward like this. Okay? And the same thing here. It's going to keep going. This is the old stuff. Now pay attention to the new information. This is the, let me choose a maybe different color. This is the DNA polymerase 3. Okay. So as DNA polymerase 3 approaches a primer, the red stuff, okay, as it approaches the RNA primer, it is replaced by DNA polymerase 1. Okay, so when the DNA polymerase 3 gets to the kind of like the junction of where the primer is, number 3 is gonna fall off, and then number 1 will carry on. Okay. And the job of number one of DNA polymerase one, I'm not gonna draw it. I don't wanna. I guess I can draw it. Like over here, right? Like you would have a DNA polymerase 3 that just falls off, and then you can have polymerase 3 coming, 1 coming up, whatever color you want to use. Okay, so this is polymerase 1 now. Okay, this is DNA. And the job of DNA polymerase 1 removes the RNA primer and insert DNA nucleotide instead. Right? So take a look here. The DNA polymerase 1 will remove the U, because U doesn't belong in DNA. It will remove the U and then change it to a T. And then here, it's A, G, and C. Those are fine, but actually they will take it off and then put it on again. Okay? They basically replace the entire primer with green letters, okay, with DNA. And that is the job of the polymerase 1. Okay. Give you a moment to copy the stuff, and then I'll show you the big picture again. So over here, take a look at these red things. Those are the primer, right? So when the polymerase 3 comes from upstream, we say, like above, it will fall off, and then polymerase 1 will replace this primer with actual DNA, with green nucleotides, so to speak, right? Same thing here. This little red part is going to get replaced. When number 3 comes from upstream, it falls off, and then that's the signal for number 1 to come on. Same thing over here. Number 3 comes, and then it falls off, and then number 1 goes on and replaces the red thing. But when we get to the very end of the chromosome, we have a bit of a problem. Okay, let me use a different color. There's too many pink stuff here already. We have a little bit of a problem here at the end. Right? The RNA primer is not replaced at the end because no DNA polymerase 3 is coming. Right? This is the end of the line. I don't have another polymerase 3 coming from upstream. Same story over here. Okay. Not replaced. Because of this problem, the ends of the DNA get shorter every time it is copied. All right, so let's say you start with DNA that's this long. When your cell divides, it needs to copy the DNA, so it gets shorter. Next time you copy, it gets shorter and shorter and shorter and shorter. Obviously, that's a big problem, right? So we're gonna look into that problem in a bit, but right now I just want you to understand why this is happening. Why are we losing DNA? Right? And again, the reason is because at the end, we don't have a DNA polymerase 3 that's coming from upstream, and nothing is falling off, so there's no signal that tells the polymerase 1 to get on. And as a result, you do not replace the primer in the end, and it just kind of disappears, and it gets shorter. I'm gonna switch back to the PowerPoint. I mean, we're basically done taking notes for all the complicated stuff. Any questions before I move on and switch back? Okay. So this is basically what I said, right? DNA polymerase 1 remove RNA primers and replaces them with the appropriate nucleotides. And then there is another enzyme that I haven't put in the note yet, but that is called a DNA ligase. To ligate is to connect. So this is an enzyme that connects the Okazaki fragments together. Right? So that happens on the lagging strand, right? Remember the lagging strand, you have all these bits and pieces of fragments, but you got to glue them together. You got to form the bond that holds the nucleotide together. That would be the job of DNA ligase. Without ligase, then the DNA would be incomplete. Okay, there will be breaks in the DNA strand. Both polymerase 1 and 3 has proofreading capability. So as it's creating the new strand, it's able to check to see if the base pairing rules are being followed, and if not, they are going to correct the error. And sometimes the error is not corrected, and that could lead to permanent change in DNA sequence, resulting in mutation. So this is a picture that shows you the Okazaki fragment, right? Yeah, there is a break, there is a break, and then the ligase is gonna basically glue them together. This is basically like if you think about this as an A and this is a C or something, it's missing the covalent bond. Sorry, it's missing the covalent bond that connects the nucleotide together. And that's what the ligase will do. The ligase will form that bond for you, so that you can have a complete continuous strand with no breaks in it. Okay, and then we only need the ligase on the lagging strand. Okay. Because you only have fragments on the lagging strand, right? So that whole thing that I showed you with all the enzymes, with the whole diagram, that's for prokaryotic replication, right? For things like E. coli, right? For us, for eukaryotic DNA replication, it's very similar, but there are three differences that I want you to kind of remember. Number one, our DNA is actually linear, okay, and very, very long. So we need to have multiple origin of replication, okay, rather than just one. Okay? We need to open up multiple bubbles, and then as it expands, it merges together, it makes the process faster. And in humans, we can have up to 100,000 origin of replications. So that's the first difference. like this, right? You got the bubble and then it opens up. We drew this before. Okay. And then number two, most of the enzymes in prokaryotes have a eukaryote version. Like, there is like helicase in humans too. But some of them have multiple enzymes representing the same thing. So as you saw in prokaryotes, we have polymerase 3 and 1, but then in humans, it turns out there are 5 different DNA polymerases. We're not going to go into the details of what they actually do. I mean, it's similar stuff, but then because it's more complicated, there are more versions of it. That's all. And then I told you some DNA are lost in each DNA replication in eukaryotes, right? I already told you at the end, because there's no DNA polymerase 3 coming from upstream, you're going to be losing a little bit of DNA every time. Now, that is not a problem for prokaryotes so much, because prokaryotes, like bacteria, they actually have circular DNA, right? So if you think about it, let's say you have the primer here, and then the primer here, like a few primers here, like they're all over the place. So when they copy the DNA though, it's gonna go this way, and then polymerase 3 falls off, replace this one, right? Same thing, like that. All right, but when you're in a loop, you circle back onto yourself, right? So for circular DNA, no shortening happens. Okay. Now, how do we deal with the situation then in humans? Like, we can't just let us lose a little bit of DNA every time we want to copy our cells. So to solve that problem, we have something called telomeres. Okay, telomeres. Telomeres are non-coding DNA, meaning they don't contain information to make proteins or anything. Okay, and they are located at the ends of the chromosomes, the ends of your DNA, basically. And it's repeats of this 6 nucleotides many times, right? TTA, GGG. It's just a bunch of them. TTA, GGG, TTA, GGG, and then repeats itself like, a couple hundred times. The purpose of having the telomeres is that they're like the little plastic thing at the end of your shoelaces. It protects the ends of the shoelace so it doesn't fray, right? In the same way that the telomeres are located at the end of the DNA to protect the ends so it doesn't break apart. And when you are copying the DNA, yes, they are getting shorter, but you are not cutting into important DNA information. You're not cutting into coding DNA. Instead, you're just cutting off telomeres. So imagine every time you tie your shoes, you cut a little bit of this plastic thing off. Okay, cut it off, right? Cut it off, cut it off, cut it off. Cut it off. Eventually, you will cut to the string, right? The shoelaces. Same thing with the DNA. If you divide enough times, you will run out of telomeres. They will get too short, critically short, we say, and then the cell will send a signal and say, we shouldn't divide anymore. Because if I divide one more time, if I copy the DNA one more time, we will actually be losing coding DNA, and that is not good. Okay, you can see in this picture, the blue X's, they are chromosomes basically, and then the part that lights up in the end, those are the telomeres, the TTA, GGG, the one that is not going to be coding for anything. All right. Now, that also creates a second interesting problem to think about. If our DNA gets shorter every time it divides, then isn't that a problem when we pass it on to the next generation? Like, let's say I decided to have a kid when I'm 30 years old, but by then my cells already have been dividing for 30 years, okay? So my DNA, when I was born, DNA this long, and then divide, shorter, right? Okay, and then now I decide to have a kid. And when I pass that on to the next generation, doesn't that mean that my child are gonna start with short DNA and then throughout his or her lifetime it's gonna get shorter and shorter and shorter, and then the next generation is gonna begin with even shorter DNA? Eventually, are we gonna run out of DNA? That's a valid question, right? Okay. Now, of course, that does not happen because of an enzyme. I'm going to come back to Dolly in a second, but there is an enzyme called telomerase. Okay, don't confuse telomeres with telomerase. Okay, the red stuff here at the end, that's the telomeres. That's uh the repeats, right? The TTAGGG, right? Yeah, and that's the plastic thing that protects the end of the chromosome of the DNA. When I say chromosome and DNA, they are basically the same thing, right? It's just the chromosome is the very compact version of DNA. So normally what happens is this. When the cell divides or when it splits, you have to copy the DNA, and then you see the red thing gets cut off a little bit, right? You lose a little bit of telomeres, a little bit at a time, okay? And then eventually, probably not like third division, like we're talking about a good, 30 to 40 division. Eventually, you're gonna run out of telomeres, and then your DNA is gonna be like damaged, right? If you keep on dividing. And like I said, at that point, the cell will not be allowed to divide anymore. However, in germ cells, does anybody know what germ cells are in the body? Like people hear the word germ, they think is like, bacteria and stuff like that. But what's germ cell in the body? Anybody know what that is? Please let me know. Mm, Uh, okay, okay. That's fine. So germ cells are cells that becomes sperm or egg. Right. So thank you, Roy. So in males, the germ cells would be in the testicles, right? They will divide to become the sperm. In female, where do we find the germ cells in female? If you have to guess. What's the female? Yeah, the ovaries, exactly, right? And then those are gonna be becoming the eggs, right? So in the germ cells, they have a special enzyme that gets turned on. The enzyme is called telomerase, okay? Telomerase. The function of telomerase is to maintain the length of the telomere. So when we make the sperm or when we make the eggs, the reproductive cells that becomes the next generation, you reset your telomeres. So when you pass it on to the next generation, they have full length of DNA to begin with. Does that make sense? Okay, so again, right, like throughout your life, your cell divides, DNA gets shorter, 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 shorter. But when you want to have kids, right, when you make the sperm and the egg, because of the telomerase, you reset the length before you pass it on. So your kids are gonna have brand new telomeres to start their life, basically. Are we together? Okay, I hope so. Now, let's go back to Dolly. For those of you who don't know what Dolly or who Dolly was, um, Dolly is the first mammal to be cloned, right, from an adult somatic cell. What is a somatic cell? A somatic cell is the opposite of germ cell. These are bodily cells, body cells. Right? So any cell in your body that's not a sperm or egg, we call that a somatic cell. Okay. Now, it's a first mammal clone from an adult body cell. And then it uses a process called nuclear transfer, which I'll explain in a second, but it only lives 6.5 years, half of the expected lifespan of the sheep. Okay, please pay attention to see this example. It will hopefully help you understand. All right, so this is how to make the dolly, how they make dolly, the sheep. Okay, so we have the sheep here. Okay, sheep here. Hey, just for the sake of asking, I know you guys don't like to answer my questions, but you know what? Answer. Say something, okay? How many sheep do you think it takes to make Dolly, to clone Dolly? It's the first mammal to be cloned. How many sheep did they use to make Dolly? Like how many parents, if you will, how many parents does Dolly had? Yeah, five? Yeah, okay, that's a good 1 and 5. Okay, yeah, good guesses, right? Okay, and the rest of you, I don't know, pick the number between 1 and 5, right? And keep that in your mind, right? And let's see. So here is Dolly, right? So this is the nuclear donor, right? So they take a cell, just a bodily cell, a somatic cell, okay, a bodily cell. Could be anywhere. I forgot where they took it from. Okay, but it could be any. Could be like um from the liver, uh from the heart, whatever. Like I don't know. But they take a bodily cell. Okay, something that's not sperm or egg, and then what they did is they extracted the nucleus. Took out the nucleus. All right. So that's sheep number 1. And then we have sheep number 2. Okay. Sheep number 2 is gonna be taking an egg. Okay, they took an egg from the this sheep number 2. This is an egg donor. Okay. And then they remove the nucleus. Okay, let's put the nucleus here. They remove the nucleus, and then that's just the shell. And here they took the nucleus, so just the inside. And what happens is they put the nucleus of the donor into the egg of the other sheep. Okay, so in the end, you have the egg plus the nucleus. Here, maybe I used a different color for the egg so we don't get confused. So like this. So this is the egg from the egg donor with the nucleus from the nuclear donor. Now, how's that different from the actual egg of the egg donor? Well, egg only has half the genetic material, right? Because look, we have 46 chromosomes in humans. Egg contains 23 and sperm contains 23. That's why 23 versus 23 gives you the 46 for the next generation, right? So here, what happens is you remove the egg, but then you replace it with a nucleus. So this egg right now is the equivalent of a fertilized egg. It's as if a sperm came and provide the other half of the genetic material, right? So now this egg has the full set of chromosome. Okay. And then we're gonna need another sheep, a third sheep. Okay. This is gonna be the sheep where you will implant the egg. What do we call people who agree to carry the baby or on behalf of someone else because maybe the other person cannot have baby of their own? Surrogate, that's right. So this is the surrogate sheep. Okay, surrogate. All right, she's happy to do it. And then when the baby is born, okay, baby, this is the dolly. So I have a question for you, okay? This dolly, is it a clone of sheep number 1, sheep number 2, or sheep number 3? Let me give the poll and see what you guys think. Okay, my question is, dolly, who took 3 sheep to make, is a clone of which one? Is it a clone of the nuclear donor, the egg donor, or the surrogate? Okay, anyone else? That's all. After all that excitement, only 6 of you gave me an answer. That is underwhelming. All right, it is what it is then. So of the people who participated, half of you say it is. Actually, that's interesting. I got 6 people who answered, but then I got like 8 answer in the end. So maybe some of you think it's a combination of 1 and 2. Actually, this is a clone of number 1. It is genetically identical to the nuclear donor because that's where all the genetic information came from, right? The egg donor is just providing the shell of the egg. There's no genetic information in it because they removed the nucleus, right? The surrogate is providing the womb for the baby to grow. There's no genetic material supplied either. All the genetic material came from the nuclear donor. So this sheep and this sheep is genetically identical. Okay. So why did Dolly only lived half the expected life span of a sheep? Well, because when they took the nucleus from the nuclear donor, that sheep has been the cells has been dividing for quite some time, right? In its lifetime, right? And so when you took the nucleus here, the DNA in this nucleus has already been shortened to a certain level. So when they transfer it to the egg and grow, Dolly was born with not brand new DNA, but DNA that has been shortened. So even though it's a baby, it's age 0, but on a cellular level, the age is actually same as the nuclear donor. Okay, so even though it's young, but biologically, genetically, is old. So that's why it didn't get to live very long, right? Because the cell doesn't really have that much potential to divide, right? Um, and that's the reason. Okay. All right. Let's shift gear and talk about telomerase and cancer. Cancer, which I'll explain in much more detail in a bit, but essentially cancer is uncontrolled cell division. Okay, the cell keeps on dividing and dividing and dividing, and the person ends up with a tumor. Okay, now tumor is not necessarily cancerous. Tumor can be benign, or they could be malignant. Malignant. Okay. When it's malignant, then it is going to be cancerous. Okay. If it's benign, that means it's not cancerous. You can just remove it. So one of the reasons why the cancer cell can keep on dividing potentially indefinitely is because of an elevation of telomerase. Remember, telomerase is the enzyme that allows the telomeres to re-lengthen before, in the germ cells, right? Okay, so you're only supposed to have telomerase in the germ cells and some kind of stem cells as well in the body, but you're not supposed to have telomerase in, like, say, your skin cell or in your liver cell and other, somatic cells. But in cancer, telomerase is switched on, right? So that's why they could reset their telomeres and they could keep on dividing and dividing, which is why one of the treatments that are being explored is telomerase inhibition. Right? They were thinking if you can block the telomerase in these cancer cells, then you can stop them from dividing, and it will help the tumor to shrink, okay, and maybe die off. So that's an active field of research, in cancer. All right. You know what? Judging from the lack of participation with the polls, I'm just gonna do this with you. Okay. What is the key difference between DNA replication in prokaryotes and eukaryotes? So let's see. Prokaryotes have telomeres and eukaryotes do not. That actually is false. It is the other way around. The eukaryotes are we are the ones who need the telomeres because we don't have circular DNA, right? Prokaryotes chromosomes have single origin of replication, whereas eukaryotic chromosomes have many. That's correct. Prokaryotes produce Okazaki fragments, but eukaryotes do not. We both do. And RNA primers are only needed in prokaryotes. That's not true, okay? Both of us needs to have the primer because DNA polymerase 3 does not know how to start the process, right? So the answer is B. Next, Okazaki fragments are joined together by, the correct answer is the ligase. And that's the glue that forms the covalent bond to connect the nucleotides together. Here's another one. RNA primers are removed by DNA polymerase 1. Okay. As number 3 approaches the primer, it falls off, and the number 1 gets on, takes away the RNA primer, replaces the U with T's, and convert the RNA into DNA, essentially. Next one, telomerase, which is the enzyme, are needed to re-lengthen shortened chromosomes in some cells. That's correct. Okay, but that there is all of the above. So I got to keep reading just to make sure. Prevents cancer development. Nope, that is not true. In fact, it's the cause of cancer, right? The cancer cell is able to turn this enzyme on and allows them to keep on dividing. So A is the correct answer. Okay. It does not remove non-coding repeats. It adds non-coding repeats of this so that you can reset the length. Right? It lengthens the ends of chromosome. Okay. So that is the telomerase. Hey, now we're gonna switch and talk about the second half of this lecture, which is the actual cell division. We know a cell, the parent cell, split and becomes the daughter cells, right? But so far we've only talked about copying the DNA. We haven't talked about how the actual division occurs yet. So that is the remainder of today's lesson. How many slides we have? We have like 30 slides. We should be able to get it done. Okay, we'll see. All right. So in the cell theory, which we learned all the way back in lecture 3, right? These are all the different parts of the cell theory. There is one part that says all living cells come from division of pre-existing cell. So essentially, that is mitosis. Right? The division of cell is called mitosis, and that's the part that we're going to be focusing on. with our lesson. Why do we need to divide our cells? What's the purpose of cell division? Well, the most obvious purpose is to replace lost or damaged cells. Right? Like if you have a cut, and then you lost some cells from the cut, you're gonna have to replace them. You damaged some cells, you're gonna have to replace them. The new cells came from the division of pre-existing cells. Growth and development, we have way more cells now than we did when we were a kid. All those extra cell has to came from mitosis. And also reproduction. Not so much for humans, okay? But for some organism, this is needed for asexual reproduction. Okay, so very briefly, if you're not sure what that means, for humans and for many animals, okay, like we reproduce through sexual reproduction. So you need two individuals, right? Okay, each contribute half of the genetic material, and then you have a baby. That's called sexual reproduction. Asexual reproduction, you only need one individual, okay, and the organisms are able to clone themselves. So there is very little genetic diversity in asexual reproduction, whereas for humans, right, like our kids don't look exactly like clones of ourselves, right, and that's for good reason. We are able to introduce genetic variation through the mixing of the genes between you and your spouse, right? So some organisms reproduce by mitosis alone. We have things like sea star. Did you know if you break off an arm from the sea star, the arm is able to grow like the whole sea star, starfish? I think you call it starfish, right? You can break off an arm and then it will go back, and then now you end up with two starfish. That's an example of asexual reproduction. A lot of these protists that we talked about, like amoeba, they produce just by splitting. Plants do this a lot of time. Like if you grow any house plants, you can break a bit off and then just leave it in water, and then it will start to create roots, and then it grows into like an entirely new plant, right? I actually was watching something over the weekend, and they were saying this never occurred to me, but like when you eat a banana, uh, there are no seeds in the banana. Okay, and that's actually because they grow bananas by cloning the plant. Okay, like they break off a shoot, and then they basically use a shoot to grow a clone of the banana plant. And because of this, the entire plantation, I think it's called, like where you grow all these bananas. All the trees are essentially clones of each other, and what they are experiencing right now is there is a virus, a fungus rather, not a virus, a fungus that's infecting the banana trees, and because they are all genetically identical, if the fungus is bad for one of the trees, it actually can wipe out the entire plantation. Because all of them does not have any genetic variation, then they're all equally susceptible to this. And they're saying, they're desperately trying to prevent this fungus from spreading and trying to find a cure. It doesn't affect human; it only affects the bananas. And they're saying if they don't find a solution soon, then banana supplies is going to plummet, and the price could go up drastically. So that's the major disadvantage of cloning. Like there's low genetic diversity, right? And then there's some other organism that's like half plant, half animal. It's called Hydra, and it grows by budding. It creates like a little mini version of itself, and then it breaks off, and then now you have a new individual. So all these only require one individual. It happens quite fast, and it requires less energy. Okay, you don't have to find a partner, and you don't have to carry your youngs. It just you just kind of copy yourself. Now, just a recap on DNA structure, so that you understand better. Here we have the DNA double helix, which is just the nucleotides, connected together with nothing else. That's just the basic macromolecules. But we know that that is actually very long and it will never fit into the nucleus. And so what happens is you have to wrap them around proteins represented here by these purple circles. Those are called histone proteins, and then you package them together. In this form is called the chromatin, right? It's DNA plus proteins. So that's the normal state of DNA in the nucleus of a cell. Okay, the chromatin. However, when we need to copy the DNA and move them around during cellular division, we need to pack up the DNA even further. We need to, create these super condensed version of the DNA. And when it's very, very condensed in its most condensed form, we call it chromosome. So like, the X that you always see in pictures, this represents a chromosome. Okay? And, at the ends, right, we now know that's where the telomeres are, right? So essentially, the chromatin and the chromosomes are the same thing, but it's just like different levels of condensation. Like how packed are we talking about, right? That's the only difference. So in humans, we have 23 pairs of chromosomes, 23 from mom and 23 from dad, and that makes up 46 in total. Now, of the 23 pairs, 22 pairs of them are called autosomal chromosomes. Autosomal chromosomes, or simply just autosomes. That's chromosome number 1 all the way to chromosome number 22. These are all autosomes. Okay, they determine basically every aspect of your body except for the sex. Okay, so like your eye color, to a certain extent, your height, right? How many fingers you have. Like you might laugh at that, but some people actually have 6 fingers. They are born with 6 fingers. I don't know if you heard that before, and that's genetically governed. It determines like if your earlobe is attached or detached, right? So all the information about you is encoded within the autosomes from chromosome 1 all the way to chromosome number 22. Your sex is determined by the sex chromosome, the 23rd pair, all right? In female, it is XX, 2 chromosome X, and in male, it is XY. So I want to ask you, who determines the sex of the child? Is it the mother or is it the father? What do you guys think? Based on what I just told you, is it? Do you think it's the mother that's deciding the sex or the father? Yeah, the father, right? Look, the father is XY. When I say decide, I don't mean like, okay, I got to choose. Like I'm gonna pass on the Y to my wife. Like, of course, it doesn't work like that. It's like 50-50, right? Okay. So there is a 50% chance that the father will pass on the X. And if that happens, the baby is gonna be a girl. If the male pass on the Y, then the baby is gonna be a boy. Okay, so it's the male that's genetically responsible for determining the sex of the child. My grandmother is really old-fashioned, and I have 2 daughters. Right? I think I told you that multiple times already. So one time she was just me and her, and she's like asking me, she's like, "Why is your wife not making any sons for you, right? It's something wrong." And then I was trying to explain to her, grandma, it's actually my fault that we're not having any you're not having any great grandsons. It's the male that determines the outcome, the sex of the child. But she would not hear it. She was like, no, it's not that way. I just know it's the female. I have 3 sons. I know, trust me, right? But despite what she said, genetically, it is the male that determines the sex. Now, when it comes to chromosome numbers, it actually has no correlation between the size of the organism and how many chromosomes they have. Humans, like I told you, has 46, but something like a goldfish has as many as 100. Okay, and a lot of the plant product that we have, they actually have multiple sets of chromosome as well. We of course, have two sets, right? Chromosome one, chromosome 23 chromosomes from mom, 23 for chromosome from dad. I keep on saying, but for some of the plants, they have like four sets. That's what tetraploid means. And then for like bread, wheat, it's hexaploid, which means it has six sets of chromosomes, as opposed to two sets in humans, right? So there are no correlation between the size and the number. It's just, it seems like a rather random uh, uh, you know uh, situation here. All right, let's talk about the cell cycle. The cell cycle is essentially the cell doing its own thing all the way up to the point where it divides, where it splits. Okay, so in the cell cycle, we have two main phases here. We have the interphase, which is where the cell spends 90% of its time in, on average. Okay? Different cell, of course, has different variability, but, generally speaking, the cell spends most of its time in interphase. This is where the cell is just performing its normal function. Like if it's a muscle cell, it'd be contracting. If it's a liver cell, it might be like, detoxifying drugs. And it's just doing its own thing, doing what it's meant to be doing. And the cell is growing, getting bigger, and preparing for the eventual division, preparing for the mitosis. The actual cell division, the mitosis, this happens like only 10% of the time. And then the parent cell becomes the two daughter cells. Okay, so interphase, mitosis. All right. Um, hold on, I forgot what I have here. Let me just like skip through a few slides and just remind myself what's going on here. Okay. So in this situation, we can see this is the cell cycle, and we have this whole purple section here. That's the interphase. Now within the interphase, we separate it into different parts as well. So we have what's called G1, S, and G2. Right. I'm just gonna briefly talk about it at this point, and then I will write down for you, later on when it becomes more important. So G1 and G2, just like your driver's license, okay? Now G1 is basically the cell growing, okay? But towards the end of G1, the cell needs to check a couple of things, right? So listen carefully. It needs to check the quality of DNA, right? Is there any mistake in the DNA? Is the DNA intact, right? Like any breaks in it? You want to check to see if the telomeres are still long enough for us to go about another round of division. Okay, so you want to check all that. You want to check if the cell is healthy. You want to check to see if you have enough resources to go through with the division. If all is well, if everything is checkbox green light, then you can move on to the S phase. If it's not good, then it's going to try to repair the DNA. And if it's not possible to repair the DNA, if the DNA is too damaged, then the cell might, be destroyed, apoptosis. Or sometimes if the cell is not, the DNA is good, but then we don't have enough resource to divide, or if the telomeres are too short, then the cell will just stay the way that they are, remain specialized, okay, remain the way they are, and not follow through with the division. okay? Because you don't want to, start copying your DNA and then find out, oh, I ran out of nucleotides, right? So that's basically G1. G1 is checking for, is the cell okay to divide? Okay, and then I told you a bunch of things that you can check for, right? So if everything's okay, then you move on, right, to the S phase. The S phase is S for synthesis. S equals to synthesis, as in like, synthesizing the DNA. And that is where you will be doing the DNA replication. Okay, you'll be copying the DNA using the method I showed you with the helicase, with the DNA polymerase 3, all that stuff. Okay, so you started with 46 chromosome. You're gonna copy all 46 of those chromosome, to have a copy. And then afterwards, you're gonna have G2. Right? G2 is basically asking, did the DNA copying, I don't know, is the DNA copying okay? Like, did you do a good job copying everything, basically? Right? Did you make any mistakes? Did you copy everything? And if it's not, okay, if it's not okay, if you made a mistake, you want to try to repair those mistakes. And sometimes if you cannot repair them, then that's it. You got to stop here, right? The cell needs to be destroyed, for example. But if everything is okay, then you can move on to mitosis. All right? So that's the interphase and the mitosis. We're going to take a short break, but before then, any questions at all? Well, if you think of any, just leave it in the chat box, and we will resume at 9:37. Okay, see you in a bit. All right, let's keep going. So let's take a closer look at interphase and what's really going on in there. So like I said, the cell spends most of its life in this stage, roughly 90%, and performing its normal function. But towards the end of this phase, the cell prepares to undergo meiosis, right? The mitosis, right? The actual division. And by the end of interphase, it would have gone through the checkpoints, right? The G1, that the quality of the DNA is good, and then it copies the DNA, and then it goes through G2 to see, whether or not the copying was successful, if you made any mistake. And if all goes well, then it's able to move on and divide. Now, this is an actual picture of the cell in interphase. And again, there was a question on the first test that's asked, to choose which of the following statement is true. And one of it says the mitochondria is orange or something. A lot of people chose that. But you got to remember, everything is actually colorless, right? In the cell because they're so thin, and they only show color because we dye them with fluorescence, right, with colors. So we can actually choose to use any color that we want. So they're actually colorless. My point is, so but there are three colors here to pay attention to. The blue shows you where um, the DNA are. Okay, and then we have the red that gives you an idea of where the cell is. It basically stains the actins in the cytoskeleton and the cytoplasm. And then we have these orange fibers that are showing the spindle fibers that are produced during cell division. Okay, so if you take a look at the nucleus here, you actually don't see much. I mean, I certainly cannot see the DNA because at this point they are not very condensed. Okay, they are still in the form of chromatin. Okay, so they are wrapped around proteins, but they're not like super coiled up just yet. Okay, so let's see. Here we have a pair of chromosome. Okay, so this is like let's say chromosome 3, right? From your dad, okay, and this is like chromosome 3 from your mom. And remember, we have 22 pairs of these things, right? 1, 1, and then 2, 2, 3, 3, 4, 4, all the way to 22 and 22. And if you're female, your 23rd pair would be an X and an X. But if you're male, your 23rd pair would be an X and a Y. Okay. So these chromosomes, we need to copy them, right? So when this is DNA replication, and in the end, you would have 2 copies of the chromosome 3. So chromosome 3, both of these would be like identical, and then you would have another chromosome 3 and chromosome 3. These ones are connected together at this location called the centromere. Okay, and the 2 chromosomes, the 2 DNA that are copied, that are exact identical copies of each other, we call them sister chromatids. Now, I want to backtrack a little bit. We'll learn more about this later on in the course, but the chromosome 3 that you inherit from your dad is not identical to the chromosome 3 that you inherit from your mother, right? For example, I'm totally making this up. Like, on chromosome 3, maybe there's a gene for eye color. Okay. So, like, let's say your dad has brown eyes and your mom has blue eyes. So you would inherit different information from each parent, right? So the 2 chromosomes are not exactly the same, but they contains the same type of genetic information. So like if this is talking about eye color, you would also find eye color on the other one, but it might not be the same version. I mean, it could be the same if both of your parents have brown eyes or blue eyes, and that then it's possible that they are the same, but they don't have to be the same. Okay? But the sister chromatids are genetically identical to each other. Okay. Now, let's move on to early prophase. Right? So our mitosis, the process mitosis actually has 4 distinct phases to it. Okay, P, M, A, T. Yeah, you can think of it's like PMET. Okay, PMET. So let's take a look at the prophase. What's happening? Okay, P stands for prepare. That's how you can remember it. You're trying to prepare the cell for the actual division. I mean, by this time, at this point, you have already copied all the DNA replication, copied all the DNA. Okay, DNA replication has already been completed. Remember, the replication happens during the interphase, right? Not during the actual mitosis. And if you take a look at the nucleus, you can see the outline of the individual chromosome. They are certainly more pronounced here than it was before. Look here, I can't even make out the outline of the chromosome because they are not condensed yet, right? And over here, it's quite starting to become more obvious. Right? So DNA replication is completed. The sister chromatids are held together at the centromere. So again, if this is chromosome 1, after DNA replication, you would have a copy of it, and it would be attached at the centromere. Now, the chromosome's got to come out of the nucleus, and in order for that to happen, you're going to have to remove the nuclear envelope. Okay, so the nuclear envelope disappears, and then the chromosome comes out. You might remember centrioles from before, from lecture 3. These are little organelles that are responsible for producing spindle fibers. And the spindle fibers are shown here in green, which helps guide the movement of the chromosome during the division. Okay? So if you look really closely at this picture, you can see the two yellow dots. The yellow dots are the centrioles, right? Like right here. That there's one here, there's one here. That's the centriole, and then all these green things are like radiating from it. Those are the spindle fibers. So that's early prophase. As time goes on, the nuclear envelope will be completely gone, completely dissolved, and then chromosomes are clearly visible now. You can make out the actual chromosome shape, and the spindle fibers that were produced by the uh, centrioles, they will attach to the centromere of each chromosome pair. Okay, so you can see these spindle fibers, these lines, they will attach to the centromere like that. So that's the end of prophase. Okay, it's basically taking the chromosome out of the nucleus, and then you have these spindle fibers that would attach onto them. So P for prepare. Next is metaphase. Metaphase. M for middle. You can remember that. So the spindle fibers, they are, again, connects to the centromere. And then there's going to be a tug of war that pulls the chromosome one way or the other until they all lined up in the middle of the cell, like this. Okay, so spindle fibers line up each pair of sister chromatids at the center of the cell, like so. A is for apart, and that's anaphase. And that's when the spindle fibers will pull the sister chromatids apart. Remember, they were stuck together like this, right? Like at the centromere, but now you split them, you pull them apart. Okay, so remember, this was like chromosome, maybe chromosome 1, chromosome 1, chromosome 2, 2, and then another 2, 2, and then 1, 1. So when you separate it, right, each half of the cell will get the exact number of genetic material, and then it will move them towards the opposite side. And finally, we have telophase and something called cytokinesis. Telophase is basically the complete opposite of prophase. The chromosome will uncoil now back into chromatin. You're gonna have to reform the nuclear envelope so that you get your nucleus once again. We no longer need the spindle fibers. They've done their job. They have already, moved the chromosome to where they need to be. So it's gone. And then the cell will split in the middle. Okay? The sharing of the cytoplasm, splitting of the cytoplasm, that's called cytokinesis. Okay? And you split, you have two cells. That's it. Now let's take a look at this picture here and see if we can make out which phase of mitosis we are looking at. Okay, let's start with something like this. Okay, this is the cell. The cell is rectangle here because we're looking at plant cells. Okay, plant cells tends to be rectangular in shape, whereas animal cells are more circular. So if you take a look at this, what phase do you think this is? Is it prophase, metaphase, anaphase, or telophase? Well, hopefully you can see that all the chromosomes are lined up in the middle of the cell, right? So this is metaphase. Metaphase. Very good. How about here? You can see the cell, and then you can see the chromosomes are kind of, separated. It's not yet at the ends yet, but they're just starting to separate. Yeah, very good. That is the anaphase. Okay, they're being pulled apart, right? And then over here, they're already at the end of the cell. And if you look really close, I know it's not the best picture, but if you look closely, you see the line is starting to form in the middle here, right? You can see a faint line, right? That's like dividing it, right? So that, of course, is the telophase. And over here, that's just prophase. Okay. These ones here, they're not dividing. So this is just interphase. I mean, interphase is not part of mitosis, but I just want to show you, right? You can't even see the chromosome. It's just like a blur, right? The nucleus. Okay. So on the quiz, whenever that is, I don't remember honestly. You might see some pictures, and then I will ask you what phase you're looking at. Right? Let's move on. So we already talked about what's the fifth picture in the fifth picture where? And the last line, 1, 2, 3, 4, 5. This one? Yeah, this one I would say I would go with anaphase, okay? Because like it's not really telophase yet because I don't really see two distinct circles like I did here, right? Um, so that's how I distinguish it. Right? Same thing here, right? You're gonna have another anaphase here, right? Another anaphase. Okay. So this is just to go through the checkpoints again with you. It's trying again. It's trying to detect. G1 is to see if you're ready to divide, and then S you make your DNA, and then G2 is checking to see if you copy everything correctly. Okay, and if there are problems that cannot be fixed, the cell will not be allowed to divide, and either it will die, okay, or it will just stay the way that it is. Okay, so just keep that in mind as we continue our discussion. Okay, during which phase of mitosis does nuclear envelope dissolves? The answer is prophase. Okay, that's when you need to take away the nuclear envelope so the chromosomes can come out, right? And move around. Oh, okay. Yeah, this is a hard question. Let me go back a little bit first. All right, let me go back. All right, you gotta do this with me in order to understand it. Okay, this is before division. How many chromosomes do you see here? It's not a trick question. Okay, just tell me the answer, the obvious answer. How many chromosomes do you see? Two, right? Okay. Hopefully you say two. If you say any other numbers, I don't know. I don't know what to tell you. Okay, there are two chromosomes here. Now, after you divide, okay, now this is the hard one. Okay, after you divide, how many chromosomes do you see here? Okay, how many chromosomes do you see? Yep. So people might be tempted to say four, right? But actually, the way that biologists decided to count them is if they are stuck together, it still count as one. Okay, so this is two chromosomes, and then this is also two chromosomes because they are stuck together. Okay, so one and two. It's only when you separate them in like anaphase, then you count them individually. Do you understand? Okay, so let's go to here now, to this question. If a cell has 8 chromosomes at metaphase, okay, so this is the cell at metaphase, 8 chromosomes. So that's 1, 2, 3, 4, 5, 6, 7, 8, right? Okay, and then you have your centrioles here with all the spindle fibers. That doesn't really matter. Okay. How many chromosomes will it have during anaphase? What's the answer, people? Is it 4, 8, 16, or 32? Okay, this is metaphase, right? In metaphase, we have 8. So how many do we have in anaphase? Well, anaphase means we're gonna separate this, right? We're gonna pull them apart. Right, so now you're gonna have one chromosome here, 2, 3, 4, 5, 6, 7, 8, so on and so forth, right? So the correct answer is 16. Okay? Have I convinced you guys? For those of you who chose 8, that is supposed to be 16? Excellent. We don't have time for this, but you can take a look at yourself, and maybe we'll take it up some other time. Okay. We have 20 minutes, and I am going to go through 2 important concepts with you. So the slides that are coming up has to do with like cancers, right? Back to the checkpoint, right? The cell cycle checkpoints. Like what's doing the checking? Like who's doing the checking of the DNA quality, and who's giving the green light or like the red lights, right? So it turns out they are mostly proteins. Okay, they're mostly proteins, and there are 2 kinds of regulatory proteins. Okay? We have what's called growth factors, which promotes mitosis. So you can think of these as like green lights. Green lights. It allows the cell to divide. It turns the cell cycle. Okay. On the other hand, we have the growth inhibitors. These are the red lights. Okay. They stop the cell from going through the cell cycle. And it could be for various reasons. Okay, maybe the DNAs are damaged, maybe we don't have enough resources, maybe the telomeres are too short. Okay, so there are a bunch of reasons why we want to stop the cell from dividing. Okay, so it's the balance between the green lights and the red light that helps control how fast the cell divides. We don't want it to divide too rapidly, right? That could lead to cancer, right? So we need to have the red lights, but we don't want to put everything on red lights all the time. If that's the case, then we can't grow, we can't heal, right? It's problematic. So it's just the right balance between the 2 that allows the organism to be healthy. Okay, so growth factors, green light, growth inhibitors, red lights, and these are proteins. Okay, that's doing the checking. All right, here is the possibly confusing part. We have something called a proto-oncogene. A proto-oncogene. Let me just switch the color back so it's not too confusing. A proto-oncogene, by the way, like we haven't really talked much about this yet, but gene is basically a segment of DNA, right? A segment of DNA, and the gene can become a protein, basically. They contain instruction to make the protein. It depends on what kind of protein you're making, right? You can mix all sorts of proteins. In the context of what we are talking about, there could be a gene that makes a growth factor protein. There could be a gene that makes a growth inhibitor protein. So the gene, the proto-oncogene, is a segment of DNA that contains the instruction to make the growth promoter, the green lights. Okay, so it normally helps the cell grow. Okay, the green light. Got it? And since the gene is DNA, and we know DNA can mutate, maybe you made a mistake when you copy them. Maybe there's too much, exposure to chemicals, to ultraviolet, right? That causes DNA to mutate, to change. So when the proto-oncogene mutates, right? Or if you got too many copies of it, and that's possible. Sometimes you copy, to the same section multiple times, that could happen. Then you end up having too many copies of it. Then the proto-oncogene becomes a bad gene that is going to be permanently turned on, or we can say permanently activated. So that's like having a green light that is always green. What happens if the green light is always green? Then traffic keeps on going, right? It doesn't stop anymore. So if you have a bad proto-oncogene that's always activated, always turns on, then the cell cycle will keep on turning, turning, turning. The cell will divide and divide and divide because it's always receiving the green light, is always receiving the go ahead to divide signal. Okay? So the cell grows out of control, which can lead to cancer. It doesn't have to be cancer. I told you, if a cell just keeps on dividing, then it gets a tumor, right? A tumor is a collection of cell, and that tumor could be benign. That's the best case scenario, or it could be malignant. Right? In that case, then it's cancerous. So the bad gene is called oncogene. Okay, so proto-oncogene, that's the normal version. Okay, that makes a green light to help the cell divide. When it mutates, it's always green. It's permanently green. Keeps the cell dividing. In that case, we call it an oncogene. Does anybody have any question? All right. The opposite is called tumor suppressor gene. The tumor suppressor gene is basically gonna make a protein that will be the red light. It will slow down cell division. Okay, it will repair DNA damage. Okay, or it could tell the cell to die. All these things are meant to slow down cell division, right? You shouldn't be dividing if you have DNA mistake. The cell shouldn't be here. It should die if it has too many errors, right? So that's a tumor suppressor gene. When the tumor suppressor gene is mutated and it doesn't work anymore, or if it's not working properly, then it's like not having a red light. Okay, the red light is not working. Without the red light, there is nothing stopping the cell from dividing. And again, it becomes out of control, which gives you the tumor. And if the tumor is malignant, it is going to be cancerous. Yeah, but best case scenario, it's going to be benign, and everything's going to be okay. All right. Now there is no different names for tumor suppressor gene when it becomes bad. Okay, we just call it like a mutated tumor suppressor gene. Whereas the proto-oncogene, when it becomes bad, we call it an oncogene. Okay. So typically, cancer arises when a mutation results in what we call a gain of function mutation in the proto-oncogene. Okay. If one copy of the proto-oncogene is mutated, it is enough to cause cancer. So this is a little bit difficult to wrap your head around, perhaps. Think of a car. Right? So the car, how do you make the car go faster? You step on the accelerator, right? Okay, so normally you can control when to step on the accelerator. But when the proto-oncogene mutates and it's a gain of function mutation, gain of function mutation basically means it's always on, right? Like I told you before, always turned on, always green light. Then it's like having a stuck accelerator. Like you press on the gas pedal and it's stuck there. It's not coming off, right? So the car just keeps on going really, really fast, right? And there's only one point of failure here. Okay, if that happens, then that's it. There's no more backup. Okay, the car will go out of control. It's very dangerous. So same thing for proto-oncogene, right? You have two genes for the proto-oncogene. So you would have basically two green lights. Okay, two green lights. Green light number 1 and green light number 2, and they go on and off depending on the situation, right? But if you have a gain of function mutation and one of them is always on, that's enough to cause havoc. Okay, because the cell see one green light, then it's like, all right, we gotta divide, and then you have uncontrolled cell division. On the other hand, for tumor suppressor gene, it is called a loss of function mutation. So again, if I may, you have two genes. You have two genes for the tumor suppressor, because we have two sets of chromosomes, right? One from mom, one from dad, right? So like you would have two red lights here. The red light would turn on and turn off depending on whether you want to stop the cell from dividing or not, right? Okay, so like if you want a cell to stop dividing, you turn on the red light. Okay, so the cell don't divide. If you have a loss of function mutation, that means the red light is not working anymore. The red light is not showing up. So let's say you bust one of the genes. So this one is not working anymore. But that's okay. We still have the other red light that's working, right? So one red light is enough to stop the cell from dividing. It's only when you mutate both copies, like if this red light also gets busted, then you don't have any more red lights and you won't be able to stop the cell from dividing. Do you understand the two difference here? Right? Over here, you have two green lights. Okay, the job of the green light is to light up and then let the cell go. But if only one of the green light is always on because of the mutation, that's enough to cause the cell to keep on dividing. Okay, it doesn't matter if the other one is work on and off like normally. If one of them is always on, that's enough to send the cell, dividing uncontrollably. On the other hand, for the tumor suppressor gene, right, same situation, two red lights, they turn on and off. But if one of them is not working, that's okay. You still have the backup. The other one can still turn on and say, stop dividing, right? It's only when both red lights are out, then the cell is going to be out of control. Any questions for me? Like, seriously? It's like really complicated stuff. I don't know if I'm explaining it properly to you guys. All right. So here is the thing, right? The second point here. People born with only one functional copy are more likely to get cancer. Okay? People who are born with one functional copy are more likely to get cancer because these are DNA and you inherit it from your parents. So it's possible that, the parents in their lifetime, they happen to have a mutation. So they lost one of the genes. One of their red light is not working anymore. They are fine. They didn't get sick. They didn't get the disease, the cancer, because they still have one functional red light, right? However, if that get passed on to the next generation, all the cells in the child is going to have only one red light left, right? Only one red light left in the child. So if any of the cell has an other mutation, that's enough to create a cancer, right? Because they only have one functional red light in all the cells. All right, so one additional mutation is gonna cause the cancer. Whereas a normal individuals with both red lights, okay, I mean there are trillions of cells in the body, right? So you can have one red light not working, okay, and then in order for the cancer to happen, you will have to knock out the other red light in the same cell before the cancer happens, right? So that's relatively rare. Okay. There's a question. Can we say cancer is mostly genetics? There is a genetic component to cancer, but it's not all genetics, as we will see later on. There is environmental factor as well. Okay, like people nowadays, there's so many carcinogens, right? They're saying consumption of red meat increases your risk of getting cancer. Exposure to sunlight, right? Not using sunscreen. Exposure to various chemicals in processed food, right? All that contribute to cancer as well. So it's not all genetics, but most cancer has a genetic basis to it. Okay, so like I said, right, could be inherited or acquired. Acquired means you get it throughout your life, right? Due to exposure to mutagens. It takes more than one mutation to cause cancer. Like what I'm showing you is an oversimplification. All right, there are many safeguards in the body that prevents cancer from happening. So it's not as simple as just having a busted green light and then cancer for sure. Like that's an oversimplification, right? Okay, but in essence, that's what it is. When someone has inherited an abnormal copy of a gene, their cell already starts out with one mutation, right? Like I told you, child who inherit the one mutation. That's why it's easier for them to develop cancer, right? They might get it earlier in life because it only takes one more hit, right, for the cell to turn bad. So that's why cancers that are inherited tends to occur earlier in life than cancers of the same type that are not inherited. There is also the factor of penetrance. Okay, penetrance is a term that is used to describe how likely it is for someone to carry a disease gene and actually get the disease. Okay, even though there is a genetic component to cancer and many diseases, uh, but genetic is not everything. Okay, think of twins. Identical twins is identical every way genetically, but they don't grow up to be exactly the same, right? Like, I mean, there's still subtle difference even in their features, right? And one might get sick and the other might not, right? So there is this environmental factor to it, okay? How you interact with the environment can change how the gene is being manifest. So that's called penetrance. If a disease has complete penetrance, it means if you have the disease gene, you are guaranteed to have the disease. That's 100% penetrance. Incomplete penetrance means even though you have the disease gene, you might not end up getting the disease. If let's say we have a penetrance of 70%, that means, it's still pretty high, or you have a 70% chance of getting it. Okay. Penetrance of 20% means even though you have a disease gene, your chance of getting the disease is very, very low. Okay. So meaning not everyone with the mutation will get the cancer. Okay. But penetrance is not just for cancer, it's like for all genetics related stuff. Now there is a gene called BRCA1 gene. BRCA stands for breast cancer 1 gene. And it is a gene that allows you to make a protein to repair DNA damage, okay, or destroy the cell if DNA damage cannot be repaired. So do you think this is a green light or a red light? In other words, is this a tumor suppressor? Is it a proto-oncogene, a green light, or is it a tumor suppressor gene, red light? If it's responsible for repairing DNA damage. Oh, it actually says on the slide. I didn't even notice. Well, because it's responsible for repairing DNA damage and not allowing the cell to go divide when there is damage, this prevents cell division. And therefore, it's a tumor suppressor gene. About 12% of women in the general population will develop breast cancer sometime during their lives. Okay, this is data for North America. About 55 to 65% of women who inherited a harmful, in other words, a loss of function BRCA1 mutation, will develop breast cancer by age 70 years. What this is saying is what I'm trying to tell you at the beginning. General public have a risk of 12%. These are people with with functioning red lights, if you would. Both copy of the red lights are working. There's only 12% chance that they will develop breast cancer in their life. But for women who inherited one non-functional BRCA1 gene, if one of the red light is not working already, then the chance of getting breast cancer by 70 years of age is very, very high, 55 to 65%, right? Because there is no backup anymore. One of the red light is already not working. It only takes one additional hit, one additional mutation to cause the cell to become cancerous, right? Some of you probably know this actress, right? Angelina Jolie. She's very famous. And one of the things she did was a double mastectomy because her mother died from breast cancer, I believe. And she did genetic screening. So she actually knows that she has a BRCA1 mutation. Right? So all her cells would only have one working red light, right? If you're following my analogy so far. Right? So that means she has a very high risk of getting the breast cancer, right? Okay. 55% to 65% as the statistic I show you. So as a preventive measure, right, she did a double mastectomy. Right? She removed her breast basically, right, so that she doesn't have to worry about getting the breast cancer. And if you're interested, you can search this up. And that actually started like a whole movement for women who who have a family history of breast cancer, that they want to know, they want to find out, right, if they are at risk. And taking some preventive measure like this could be an option for some people. Of course, it's not an option for everybody. It's expensive, right? These kind of surgeries, it's a high risk, but it doesn't mean you are certainly going to get it because of the penetrance, right? And it's an individual choice, right? That people can make. So that's the BRCA1 gene. All right. I think that's a good place to stop. I mean, we just have a couple more slides to go, but we can certainly do that next time. It's already very, very intense. Now, lecture 7 requires some problem solving. Okay. So I don't know how you guys are doing these lectures with me, but you should print out the study guide for next class. Okay, I know I haven't really been using the study guide too much just because we don't have enough time, right? But then I do post the answers for you guys to look at. But for lecture 7, you need to print out the study guide, right? Because there are these questions that we have to do together in class to help you understand it. Okay, these are kind of like math and chemistry problem solving. So make sure you get a copy of that. And that's it. Okay, I will see you guys next next Tuesday. All right, have a good one. Bye bye.